All right. So yeah, today I'll be talking a bit about a big project we've had to sequence the Solanum lycopersicoides genome and assemble it, and then use it for some interesting biological stories. But before I go into that, I wanted to thank all of the numerous people that have been involved in this project. So as you probably know, genome projects tend to involve a lot of people. So this has been a great team to work with, especially Adrian Pau and Bjorn Usedel. We had a big push yesterday to finally get this paper submitted. So that was pretty exciting, and they were instrumental in getting that accomplished. And real quick, I just wanted to tell you a little bit about where I work. So I'm at the Boyce Thompson Institute. So the Boyce Thompson Institute is on the Cornell University campus. So we work very closely with Cornell, but we are an independent institute. We have, I think, 18 faculty members now that work on a variety of topics, mainly plants or things that interact with plants, or the research focuses. So there's a lot of work on tomato and other Solanaceae. There's a lot of work on pathogen response, and there's also several computational groups. So if anyone, probably a number of you in the audience, use the Sol Genomics network. So that's actually run from Lucas Mueller's lab, which is at the Boyce Thompson Institute. All right, so on to lycopersicoides. So probably many of you are familiar. There's a number of wild species that are close relatives of tomato. So in the tomato clade, altogether, I think there are 13 species. So this tree shows some of them. I think Huila sensei, I don't know if I'm saying that correctly, is one of the species that's not in the tree. So there's red-fruited species, so that would be Pimpinellifolium and the cultivated tomato. There's two orange-fruited species, Cheesemania and Galapagense, which are found in the Galapagos Islands. And then the remainder of the species are green-fruited. And then lycopersicoides is more of an outgroup, and under the right circumstances, it actually has purple fruits, which you can see in the picture there. And these species are all found on the western side of South America and the Galapagos Islands, as you can see on the map there. So some of the nice things about having these wild species is that they can actually be crossed with tomato. I'm sure many of you are aware, which is really nice because, of course, tomato went through a number of bottlenecks during domestication. So it came up out of South America to Mexico. Then it was brought over to Europe by the early explorers. And so most of the tomatoes we have kind of went through this sort of Z-shaped travel that you see on the map there. So cultivated tomato has less diversity than the wild species. And so we can use the wild species to introgress important traits into cultivated tomato. So things like abiotic and biotic stress tolerance, fruit quality traits, and other agriculturally useful traits. And I always use this as an opportunity to show off my tomatoes. So I'm a big fan of heirloom tomatoes. Anyone that knows me has had the pleasure, I hope, of hearing about them. So this is a picture of some of my tomatoes from my garden a couple years ago. And I guess I was bored that day, so I decided to arrange them like a rainbow. So you can see there looks to be a lot of diversity based on phenotype, but much of what you see here is just controlled by a handful of genes. So again, this is what makes the wild species really, really useful. So one of the wild species, which I mentioned, which is more distantly related, is Solanum lycopersicoides. So it's been used to create introgression lines. So there's a paper from 2005, Kennedy et al., that describes these lines. And it's a cross between VF36, so cultivated tomato, and then LA2951, which is a lycopersicoides accession. And in this figure here, you see the 12 chromosomes of tomato, and there are traits, so fruit quality traits marked in red and orange. So red means that there was a increase in carotenoid accumulation, and orange is a decrease. And then also on chromosome 4, you see this little blue blip. So that area is involved in resistance to Pseudomonas syringae, which I'll talk a bit more later. And there's actually kind of a cool story behind this. So Ari Feder, who was a former postdoc in Jim Giovannoni's lab, was growing these lines out in a local field here in Ithaca, New York, where, as you can imagine, it's kind of cool and wet, which are excellent conditions for bacterial spec. And he went to the field where he was growing the ILs, and he found that most of the plants were decimated by this disease. But there were a couple ILs that seemed to be just growing along just fine. So you can see the plant on the left side here looks pretty healthy. So that was one of the ILs. And it turns out these ILs all had this introgression from lycopersicoides on chromosome 4. So of course that was something that became interesting to Greg Martin's group, and they went on to find the locus that is involved in that resistance. And I'll talk a bit more about that in relation to lycopersicoides in a bit. And then in this figure A, you can see the difference in carotenoid accumulation in the different IL lines. And just keep in mind, this is a pretty wide cross, so it's kind of difficult to do, I believe, because cultivated tomatoes is fairly distant from lycopersicoides, since it is more of an outgroup. All right, so we, for the reasons just described, thought it would be nice to have a lycopersicoides genome assembly so that we could explore some of these biological questions. So you can see a little picture of lycopersicoides there. That is not the one we sequenced, but that is one that we did have growing in the growth chamber at BTI. The one we sequenced was actually out of Björn Usedel's group. So we sequenced it with PacBio. I think we had more than 80x coverage. And then we tried a few assembly approaches to see which one was going to give us the best results. So we assembled it with Falcon and Canu. It turned out that the Canu assembly was a bit better, so we moved forward with that one. We used some Illumina reads to polish the genome, try to get rid of any residual indels that might be there from the PacBio sequence. Then we sent it off to Dovetail to do Hi-C so that we could get the contigs up to pseudomolecule level. And we filled in the gaps that were created from the Hi-C with a tool called PB Jelly using the long PacBio reads. And this is how our assembly looks. So it's pretty decent. There is a newer version of the tomato genome now that's a bit better. So I probably should have included that in this chart. But anyways, it gives you some idea of the quality of this assembly. So we got it up to pseudomolecule level. So there's 12 pseudomolecules. We have, I mean, it's basically chromosome scale whenever you look at the longest sequences. The contig N50 was about 253 kb. And the total length is pretty close to what we would expect the Solanum lycopersicoides genome to be. So that was good. That means that we likely captured most of the genomic sequence in our assembly. And then about 10% of the assembly we were not able to anchor with the Hi-C data. And you can see that our Busco metrics are comparable to both the Pinelli and the tomato assemblies. We do have a higher duplication rate in this assembly because it is an outcrosser and it has a high heterozygosity rate. So there's some duplication in our assembly. And there are tools that we can use to remove this. So next we looked at synteny. So this is work that was done by Adrian Powell at BTI and Asha Duhan, who was an intern at the time at BTI. And so we compared lycopersicoides to cultivated tomato and then also to Pinelli. And so these charts are really pretty to look at, but I guess they're not really all that exciting because they're showing us kind of what we would expect, that there's a high degree of collinearity between the lycopersicoides genome and both of these fairly close relatives. So nothing too shocking there. We also, this kind of gives you a little bit of a finer view of what that synteny looks like. And we did find a couple of regions where inversions that were previously noted were found. So we found something on chromosome 4. So there's a little blip there, which is an inversion. And then there's also an inversion on chromosome 10. And we tried to confirm the remainder of these, but it was a little bit tricky. So it is possible that a couple of the little blips you see could be some residual errors from the high C scaffolding, because sometimes it doesn't quite get the orientation of things right. So then we took so there's 71 IL lines from the 2005 paper that I mentioned. So we used RNA-seq data from all of these IL lines to map where the introgressions are. So that's what this is showing. Each little track is a different IL line. And then the blue and the green designate whether that region was heterozygous for the introgression or homozygous. So you can kind of get a rough idea of where these introgressions are. And this is all based off of the lycopersicoides genome in this particular figure. We also have that information based off of the cultivated tomato as well. And for anyone that's interested in these, we do have like a giant tab delimited file that says specifically where all of these introgressions are. So this is a really nice resource for people that might be interested in exploring biological questions in lycopersicoides. One utility of having the lycopersicoides genome is that it allows us to better analyze RNA-seq data. So you can see here in this graph that whenever you map, say, lycopersicum RNA-seq to lycopersicum, so mapping it to itself essentially, you get a pretty high mapping rate of the RNA-seq. But this mapping rate goes down as you increase divergence away from cultivated tomato. So Pinelii, you can see it maps a little bit less, and then we get the lycopersicoides, and it's dropping below, looks like 75. So if you're doing things like RNA-seq, you're probably not getting very accurate information for expression, for your genes of interest if you're doing something like differential expression. And there also, another problem is that lycopersicoides may have genes that aren't present in cultivated tomatoes. So you're completely losing the ability to detect any information from those genes. So this was some work done by Lance Courtney and Jim Giovannoni's lab. So he basically has made a tool that takes into account where all of these introgressions are, and then it just subs in the lycopersicoides genome, so that whenever you're analyzing RNA-seq from the IL lines, you can create a genome that reflects the genome that IL line actually has. So this increases your ability to map RNA-seq and also to detect differential gene expression. So the next thing we did, of course, was we ran some annotation, because everyone wants to know where the genes are in the genome. What good's a genome without knowing where the genes are, I suppose. So we used a pretty standard pipeline for that. We did repeat masking with repeat scout, repeat modeler, and repeat masker. So that, of course, does some de novo prediction of repeats and makes a library that you can use in repeat masking. Then we also had RNA-seq and protein evidence. We also had iso-seq from a variety of tissues that were pulled. So iso-seq is long-read sequencing, which is really nice for annotation because you have an entire transcript, so it allows you to more accurately call gene models. So we had that data, and we used all of this as input to Maker to predict gene models. And then Bjorn's group ran his Mercator pipeline to do some functional annotation. So what are those gene models? Now we have the gene models. What do we think they might be characterized as? The other thing we have is a WebApollo for this. So we did some manual curation specifically of some genes that were involved in immunity, because we, of course, were interested in that, since lycopersicoides has some novel pathogen response. So WebApollo is, I think it's actually just called Apollo now, is a nice tool. So it basically works with JBrowse, and it allows you to edit gene models. So you can load RNA-seq data, iso-seq, whatever evidence you have as tracks, and then you can look at that supporting data to figure out if the gene model looks to be correct or if there are some problems. So this is nice whenever you have a specific group of genes that you're interested in making sure that the gene models were called accurately. Okay, so then Adrian did some work on classifying the repeats. So about 68% of the genome sequence that was contained in pseudochromosomes was found to consist of repeats. And of all the repeat classes, the long terminal repeat retrotransposons was the largest proportion of repeats, and it was about 57% of the lycopersicoides genome. And also this is found in this, the numbers aren't exactly the same, but you also find a large proportion of these in cultivated tomato and pinelii. So that's just kind of in parallel with that. And I'll come back to this a bit more in later slides. To give you an idea of what our annotation looks like, here are a few stats on that. So we have slightly more gene models in lycopersicoides and Heinz, but they're fairly similar. And overall, things are looking pretty close to Heinz, which is good because Heinz is our model species, and we know a lot of time and effort has been invested in that genome. So the fact that it's looking kind of comparable to that, I think, is a good sign. And you see that we still have a higher level of duplication. So again, not surprising since this is a fairly heterozygous species. But we are able to get most of the Busco set. So these are genes that should be present in these species. So we're able to find most of them in our annotation as we did in the genome. So that means our annotation is probably pretty decent. So here we have another pretty circos plot. So here we're looking at gene density on track A, and B is the repeat density. And you can see that they're sort of inversely related there. And you see very similar thing in the tomato genome as well, where the gene density is a bit higher on the chromosome ends. And then whenever you get into the centromeric regions, there's less genes and more repeats. So probably not so surprising. So of course, you have to have a gene family analysis in a genome paper. So that's what we're looking at here. So this is just basically running OrthoFinder with each of these 5 species and seeing what we find. So there was a total of 28,833 gene families that contained 193,788 genes. And you can see that there's a large portion of them there in the center that are overlapping, which we would expect. We also see 481 families that are unique to lycopersicoides that contain 1,696 genes. So it's possible that these could be weird things with the annotation, but they also could be biologically relevant. So they could be things involved in some of the neat phenotypic things that we see in lycopersicoides, sort of what makes lycopersicoides lycopersicoides. We did a CAFE analysis. So this is looking at lineage-specific expansion and contractions of gene families. So the numbers in green means that they have expanded, and the numbers in parentheses are significant, the number that were significant. And then the red is the numbers that contracted along that lineage. And so whenever we looked at the gene families that were expanded along the lycopersicoides lineage, we found that they often were involved in things like pathogen and stress response, also self-incompatibility, and there were a lot of transcription factors. The contracted ones were cell wall related, so whatever that means. So this was work that was done, I think, mainly by R.A. Fetter. So I mentioned at the beginning of the talk that lycopersicoides has purple fruits. So why is this? So it's something partially to do with carotenoid biosynthesis and some other things that I won't address here. This isn't exactly my wheelhouse. So you see the pathway for carotenoid biosynthesis on the left, along with the key genes that are involved in that pathway in blue. So whenever red grapefruits are known to have a decreased expression of the CRTL genes, which you can see sort of at the bottom of that pathway. which results in accumulation of lycopene. So what happens in fruit that are non-red ripe? So if you look in figure B, you can see the red ripe on the left and the non-red ripe on the right. And what we're looking at are expression values. So these are FPKM values. So the gene expression in the lycopene accumulating cultivated tomato and also pimpinellifolium is so on the right, you can see that the upstream pathway genes are more highly expressed than the species on the right. But then whenever you get down to the CRTL genes, they tend to be more lowly expressed than the species on the right. So this kind of explains some of why we're seeing this different fruit color. And Ari did find a previously uncharacterized gene, so this CRTL-B3. So if you notice in lycopersicoides, it's pretty highly expressed compared to the other species. So he went on to do a little bit more work with this. So he looked at the lycopene to beta carotene ratio, which you can see in the blue bar graph there. And he found that it's and the three that are on the right are ILs that are containing regions that are relevant to this phenotype. So you can see that they have a lower ratio in comparison to VF36, which would be a red ripe fruit. And then he was able to go on, and I'm not showing this data, but he was able to show that it is actually able to convert lycopene to beta carotene using E. coli. I mentioned before that lycopersicoides has some pathogen resistance, but it turns out that, so this is resistance to Pseudomonas syringae pathovar tomato race 1. So it has resistance to that, which is pretty cool because I don't think there's been any genetic resistance for this strain detected before. But so this was work by Emma Jobson, who was a former postdoc in Greg Martin's lab. She found that lycopersicoides, at least LA2951, is susceptible to PST race 0. So this resistance is conferred by the genes PTO, PRF, and FEN. So in this experiment, she inoculated these 6 different varieties of tomato with DC3000, which has the effectors AVR-PTO and AVR-PTO-B. So both of these are recognized by the PTO-PRF complex. So the controls are the 3 species the 3 lines on the left. So we have RG-PTO-R, so it is resistant to race 0. So you can see whenever it's inoculated with DC3000, you don't really see anything happening. That looks like a nice healthy leaf. The next 2 lines have a non-functional PRF and a non-functional PTO. So both of these genes are required for the resistance, and you can see that both of these leaves look pretty susceptible. The next 3, so the last three lines, these are all IL lines, which have introgression from lycopersicoides in that chromosome 4 region that I showed you way back at the beginning. And so all of these look to be susceptible as well. So Emma went on to look at what might be happening in the genome. Since we have a genome and annotation, she was easily able to do this. And you can see that LA2951 does look pretty different from RG-PTO-R and also from Heinz. So Heinz is also susceptible. So whenever she did this work, she found that PTO and also the family members PTOC and PTOF were missing in lycopersicoides. It does have a PTOD, but it contains a premature stop codon. She did find a homolog to PRF, but this was also likely non-functional because it had an insertion with a mutation that led to a premature stop codon. So it seems likely that the susceptibility of the ILs to race 0 is likely due to a non-functional PRF and or PTO gene in LA2951. So Adrian Powell did some work to look at the basis of stress tolerance, so abiotic stress tolerance in lycopersicoides. So he coupled bioclimactic data with collection locality data from TGRC. And he looked at cold and at precipitation, so cold on the left and precipitation on the right. And he found that overall, at least based on this data, that Solanum lycopersicoides is exposed to more extreme climactic conditions than lycopersicum. And so for the minimum temperatures in the coldest month of the year, all species were significantly different from each other. And lycopersicoides was subjected to the coldest temperature. And then in the driest quarters, both lycopersicoides and Pinelii received precipitation that was significantly less than cultivated tomato on the left. So you can see that in the graphs there. So this was pretty cool, and he wanted to see if these results correlated with long terminal repeat abundance, which was seen in the Pinelii paper. They had looked at this, so we wanted to see if something similar was going on for lycopersicoides. So you can see here that the type and abundance of long terminal repeat elements in lycopersicoides, that was determined using LTR retriever with inputs from LTR harvest and LTR finder. So he looked at lycopersicum, Pinelii, and lycopersicoides. And he was able to confirm, as reported previously using this pipeline, that Pinelii had a greater abundance of younger LTR retrotransposons than cultivated tomato. And it's particularly pronounced for copia type elements. And you can also see that lycopersicoides tends to look more similar to Pinelii. So sort of correlating this with some expression data. So here we're looking at some drought responsive genes in Solanum lycopersicoides. So this was based on RNA-seq data. So water was withheld and expression was quantified at 2 time points and a control treatment. And in doing this, he was able to find 53 drought responsive genes for the first time point that were correlated with nearby LTR TEs. And then at the second time point, there were 17 genes with expression correlated to LTR TEs. And there were 12 genes that were shared across both of these time points. And I should mention this Marcus McHale was also a big part of this work as well. So you can sort of visualize, this data is all available. I don't know that it's publicly available yet, but it should be very soon because we just submitted the paper on all of this. But if you do go to the Sol Genomics site, there's this really cool tool, which I'm displaying here, that you can load expression data into, and it allows you to look at correlations between genes. It allows you to visualize sort of the expression data in this Rubik's Cube looking thing. You can move things around. It's a really cool tool, so I highly recommend you check it out. Yeah, and so as I mentioned, we just submitted this work yesterday, so fingers crossed we get some good news. There's a preprint on bioRxiv, but the submitted version of the paper is pretty different from that one. I think we do plan to update that. So if anyone's interested, let me know, and I can give you that information. And we did make the genome available pre-publication. So if you're interested in that data, you can go to Sol Genomics and check it out there. All this stuff will be loaded into GenBank. I think that's actually happening today. So if anyone's interested in any of the raw data, the PacBio, the RNA-seq, or any of that, that will all be available in GenBank very soon. So summarizing this work, Solanum lycopersicoides has had significant expansions in pathogen and stress response genes, alpha compatibility, and transcription factors. It accumulates significantly lower levels of lycopene than red-fruited tomatoes, partially at least because of more highly expressed CRTLB3 gene. And it also likely lacks functional PTO and PRF genes. Solanum lycopersicoides has a greater abundance of younger LTR copia elements than cultivated tomato, and that could play a role in the resilience of lycopersicoides. And all in all, we think that our reference genome has been pretty instrumental in allowing us to explore these biological stories. So it's always nice to have a reference genome in the species that you're exploring. So that's it for lycopersicoides. Real quick, so in my introduction, it was mentioned how I'm the director of the Computational Biology Center at BTI, and I just wanted to point anyone in the audience that's interested in doing bioinformatics. We develop a number of tools, do workshops, and we also provide a lot of help. So if you're interested in assembling a genome or doing some RNA-seq or machine learning is kind of the new thing that we're venturing into, we do have tools available. We've worked closely with Cyverse to do some workshops, and some of these things are available on YouTube. You can find most of our tools on GitHub, and also the tools that are used as sort of demos in our workshops. We have an intro bioinformatics course, which you can get to off the Boyce Thompson Institute website. So we're here to help. That's sort of our goal is helping democratize bioinformatics. So any questions about anything, please reach out. We love to help. And here's all of the wonderful people that I get to work with. So they're a great team, and it's a delight to work with them. And I think that's all. If you have any questions, please feel free to ask them. I will try to answer.
Thanks. Yeah, thanks, Suzy. That was a cool talk. If you have questions, you can either just type question in the chat, and then I'll call on you, and you can turn on your camera or microphone and ask it. Or if you want to type the whole question, that's also fine, and I can read it. It looks like Zachary Littman, you have a question.
Hi, Zach.
Hi, Suzy. Good to see you.
Good to see you.
Thanks for the talk. I may have missed it, but I'm wondering about the expression analysis in the lycopene biosynthetic pathway. So there you were showing an association with expression level difference in the ratios as it relates to the fruit color variation.
Yeah.
But this relates to a larger question. Have you done a global structural variant analysis, and in particular looking at whether any structural variants might be influencing or associated with the expression variation that you're seeing between those two species?
Yeah, that would be really interesting to do, and we have not. The structural variation analysis we did was pretty large, like global, just looking at sort of larger rearrangements, but we haven't really done any sort of like micro, I guess, looking at that smaller scale. So yeah, that would be really cool to look at. We should do that.
Thanks.
Looks like we've got a question from Stacy Smith, so I can read it. I'm wondering if there is a plan for which wild tomatoes to make reference sequences. I'm just guessing the plan is eventually to do more across the clade. And thanks, Suzy, for the interesting talk.
Hi, Stacy. Thanks for attending. Yeah, let's do all of them, right? I think that we've submitted in a couple of proposals sequencing more wild species, so doing de novo assemblies. There's a lot of sequence out there, but I think a lot of the genomes that had been sequenced, it was more resequencing and not standalone de novo assemblies. So I would love to be involved in assembling more genomes or things outside of the tomato clade too. It just makes, I think it's so much nicer when there's a genome available. It kind of opens a lot of doors, I think, for questions.
So we've got a question from Pat Bettinger, so go ahead.
Hi, Suzy.
Hi, Pat, how are you?
Good, good. And you probably already know what my question is. What was the self-incompatibility? What was the expansion you saw in self-incompatibility genes?
Yeah, I'll have to look back at that. I think that really stuck out to me too, of course. So I was, just kind of doing some functional analysis of the gene families, and I didn't really go beyond that. I just saw that there were some families that were characterized as self-incompatibility related that had expanded. So yeah, reach out if you have some ideas and you'd like me to look more into something. I can certainly.
Oh, I definitely will. Yeah, definitely. Thank you.
All right. I'm not seeing any other questions. Anybody else? Okay. Well, thank you, Suzy. That was super cool. So, oh wait, I see something. Federico has a question. Go for it.
Hi, great talk. Yeah, I have a question about something that you probably didn't want to mention about the fruits, the purple fruits.
Mm-hmm.
Are anthocyanins involved in those fruits in that coloration?
I believe so. And just to be really honest, fruit pigment is not, as I said, my wheelhouse. So if you have more specific questions, I'd strongly recommend you reach out to Ari Fetter, who did that work.
All right. And then more related to the photo that you showed about your tomatoes in your garden. Some of those are purple. Are these Solanum lycopersicum or?
So they all should be, and I have not sequenced them to do any sort of confirmation, but all of the tomatoes in that picture should be heirlooms, which should predate any sort of modern breeding program. But I mean, it's possible that something crept in there and you know, I just haven't really explored that. If I had the money, I'd sequence everything.
No, it's just that I've heard that the cultivated tomatoes lost the anthocyanins in domestication, but they are present in fruits of other species. So I was wondering if it was an introgression or if it was not true that all domesticated tomatoes have lost
I don't know conclusively, but as I said, they shouldn't have introgressions, but that has yet to be verified.
All right, thank you.
Looks like we do have another question from Pablo Perez.
Thank
So
you.
Go for it.
Thank you, Susan, for your talk. I have a question about the assembly.
Yeah.
Recently, I've seen some projects where people do hybrid assemblies using different approaches, like Illumina and PacBio.
Mm-hmm.
Do you think it has some kind of impact in the final assembly or some kind of advantage?
Yeah, so we actually do that too. We have one of those little nanopore MinION devices in my office. And it's nice because it just, you know, kind of liberates you to sequence whatever in your wherever you want because it's so small. So for those assemblies, we usually, if we're just doing sort of a draft assembly, we'll use Masurka to do a hybrid assembly. So using Illumina data and nanopore, and we'll combine it. So it allows us to do pretty cheap genome assemblies. I think though, if you want something really high quality, I would go with the PacBio Hi-Fi. That's we're actually trying that out with a couple polyploids right now. And what's nice is that it's very high quality, and also the assemblies run much quicker. One of the problems that we find with Masurka is that for the large genomes, so I actually work with Stacy Smith on Iachroma, and that genome assembly ran on an 80-core server. So it wasn't a cluster, but it still had 80 cores. It ran for I think it was 4 months before it completed, and that was a hybrid assembly. So that's one of the things to be aware of when you do hybrid assemblies is the larger the genome, it really can take quite some time unless you have a huge cluster or something to run it on.
Okay, thank you.
Mm-hmm.
All right, got a question from Gustavo Silva. Go for it.
Hello, thank
Hi.
you for the talk.
Mm-hmm.
I just jumped because you got into the topic of the assembly and the PacBio. I am close to decide like some new sequencing on PacBio. I'm wondering if you can share a little bit of your experience about like the minimum depth that you would suggest to get decent assemblies using the PacBio, especially the Hi-Fi.
Yeah, so the assembly I talked about today was using older PacBio data. So I think at that point in time, it was recommended to have at least 50x. But as I said, we're just kind of venturing into Hi-Fi. So I personally don't have any results yet, but my collaborators are using like 30x Hi-Fi and they're getting great assemblies with that. So I think you can get away with, you know, a bit lower coverage whenever you're using the Hi-Fi. And as I said too, the assembler for that is much more efficient as well, which is nice. He said they can run assemblies in a couple of days.
And it's supposed to be like the accuracy will be way better. Maybe we can, we need less polishing, and maybe the Illumina sequencing should be like less truthful.
Yeah, so our current assemblies, we always polish with alumina because we find that even whenever we do polishing with the long read, there's still some residual indels that don't get corrected, which is bad because then it breaks your gene models. But again, I haven't really explored high-five results. But if you want to reach out to me whenever I start, we should have some data like in the next couple of weeks. So I'll have a better idea then if you want to chat more. And I thought I saw Ari on the conference too, so he's another good person to ask about genome assemblies.
Great, thank you.
Okay, we've got a question in the chat from Kevede Muleta. I was wondering if those genes that are unique to the lycopersicoides genome belong to certain functional groups.
Yeah, so in the gene family analysis, the ones that were unique, yeah, I think they probably do. I can't remember if we looked at what those were specifically. Probably one of us did, but I can't recall off the top of my head what they were. But yeah, if anyone's interested in that, just please reach out and we can certainly look at that. A lot of times too, what you'll find whenever you do gene family analysis is the ones that are specific to a species sometimes can just be artifacts of the annotation too. So maybe something that's like a repeat or something didn't get filtered out. So sometimes those things aren't necessarily biologically interesting, but some of them are, I'm sure.
Cool.
Mm-hmm.
All right. Any other questions? I'm not seeing. Nobody's typing.
Am I off the hook?
Yeah, I think so. Okay, thanks again,
I don't want any
Suzy.
more quality questions.
Oh, thanks. Yeah, that was great. So, and the talk will be up on YouTube at some point soon, probably later today. And I guess before everybody goes, or whoever's still around, next week we're going to have Andrea Berardi, and she's going to talk to us about reacquisitions of pigmentation in petunia. So hopefully see you all next week as well. All right. Thanks, everybody.
Thank you.